The overall goal of this procedure is to discover novel extracellular protein interactions involved in cellular recognition processes, in particular, membrane-embedded receptor proteins. The AVEXIS approach is designed to circumvent the very weak interaction affinities that typify this class of protein interactions, and the fact that membrane proteins are difficult to biochemically manipulate. This is accomplished by first compiling a recombinant protein library of the extracellular regions of cell surface receptors. This is achieved by designing expression plasmids to produce the ectodomain regions of the receptors as soluble fragments as both monomeric biotinylated baits and pentamerized enzyme-tagged praise. The second step is to express and normalize the proteins to be ready for interaction screening. All the proteins are produced in mammalian cells to ensure that post-translational modifications such as disulfide bonds and glycosylation are added. A crucial step in the protocol is to ensure that the bait and prey proteins within the library are all normalized to within threshold levels. Next comes the exciting part, screening for novel interactions. The baits are arrayed into individual wells of a microtiter plate and then probed for interactions using the praise. Then a wash step is performed. The final step is to add the substrate for the enzyme to detect any captured praise on the arrays of bait proteins. Ultimately, the results of an AVEXIS screen can show which cell surface receptors are able to form binding partners and may therefore be involved in the recognition and communication processes between cells.